Myself, Dr. Raktim Mukherjee, Assistant Professor and Head of the Department, Shri P. M. Patel Institute of PG Studies and Research in Science, affiliated to Sardar Patel University, Anand. The title of my today's talk is Alternative Pathway of Complement System. For all those who joined late or has not gone through my earlier videos or online lectures, for them, in the first segment we introduced to complement system, the various players of complement, the way how complement functions, especially the notations that we use for the active complement components with a bar. In the second segment, we talked about classical complement pathway, and now in the third segment, we shall describe the alternative pathway of complement system. As the name suggests, alternative, the very name suggests it's not the original pathway, it is an alternative pathway, and this alternative pathway is initiated by several factors such as bacterial endotoxin, the polysaccharides, the polysaccharide capsule, even some of the aggregates such as immunoglobulin E and properdin. So, I will directly come to the initiation part. This pathway is initiated by bacterial endotoxin, polysaccharide capsule, aggregates of immunoglobulin E and properdin. This pathway is relatively shorter. Do not be very excited, because very soon I will tell you the sad part of it, that it is not actually that shorter as you think. It starts with C3. In this pathway, the steps such as C1, C4 and C2 are bypassed. So, directly you have C3 followed by C5, C6, 7, 8, 9. The steps C1, C4 and C2, which was very prominent in classical pathway, those steps are bypassed as far as alternative pathway is concerned. Very important point often asked in Sardar Patel University and other CSIR net entrance examination: antibodies are not required to initiate activation of this pathway. Therefore, alternative pathway can be said as a true representative of innate immune system, because antibodies are not required to initiate activation of this alternative pathway. This pathway also provides a means of non-specific resistance, added layer of protection, or non-specific resistance, since antibodies are not necessary for alternative pathway. Now comes the, I said that do not get very excited, something I will discuss. You know, we all know that the steps C1, C4 and C2, they are bypassed. We all know this. But now there was a research paper that came up in 2008, and in the year 2008, probably, not probably, it was certainly, myself and Milan sir, we were teaching the first batch of our masters, that is your Jayesh sir's batch and Akash sir's batch. At that point of time, the research was going on. So, that particular batch as well as the next batch, Megha madam and Sonal's madam batch, they did not study this fragment that I am going to teach now, that is your batch. It came many years later, in fact, because at that time alternative pathway was very short, it did not have C1, C4 and C2. Now, according to the latest research I have included in my presentation, the alternative tickover pathway, alternative initiation of alternative pathway, now even the initiation is done by 3 separate ways, and they are the alternative Tickover pathway, the alternative properdin mediated pathway or properdin activated pathway, and alternative protease activated pathway. This leads us to a contention where a possible essay type question can be is very important, not must be, can be asked in examination, and that is discuss the three distinct ways by which alternative pathways get initiated. If it is asked in 2 mark, Tickover pathway, properdin activated pathway, and protease activated pathway. If it is asked in 6 mark, 3 paragraphs, 1 paragraph each on Tickover pathway, properdin pathway, and protease activated pathway. Of course, lengthiest is Tickover pathway, rest 2 are just few sentences. So, now students try to understand even alternative pathway can be initiated by 3 means: the Tickover alternative pathway, the properdin activated alternative pathway, and protease activated alternative pathway. We start our talk with Tickover pathway. I just give you the example of those Tic Tac chocolates that are available. You just keep your thumb, open it, and then pop it into your mouth. So those examples, Tickover, every time it keeps on ticking, just like your wall clock. Even during the dead night, sometimes everything is so silent. During winter, your AC is not running, you don't have any other external sound. Sometimes you can feel the movement of those clock hands. So alternative Tickover pathway, the alternative properdin. So Tickover pathway utilizes 4 serum components, and those 4 serum components are C3, factor B, factor D, and properdin. These are the 4 serum components that your Tickover pathway utilizes. Why I am giving this detail, student, is because Sardar Patel University or any other university can ask this as a 2 mark question. Simply enlist, no need to describe, enlist the 4 serum components that are a part of Tickover pathway. Your answer should be C3, factor B, factor D, and properdin. We continue our journey further. When you talk about the initiation of alternative Tickover pathway, initially C3 was found to be in very high concentration in the serum. C3 was in high concentration in the serum. This C3 undergoes spontaneous hydrolysis in its internal thioester bond. This spontaneous hydrolysis at its internal thioester bond hydrolysis. So, C3 undergoes spontaneous hydrolysis at its internal thioester bond. Because of the spontaneous hydrolysis at its internal thioester bond, this C3 yields a molecule, and that molecule is a conjugation of C3 with water called as C3H2O. For many years, scientists never understood that this C3H2O is such a suspense molecule or wonder molecule that it will open up a separate Tickover pathway of complement system. Why? Because I would like to pass on this information to students. So, they did not use a very simple instrument, spectrophotometer, and when they used spectrophotometer, to their utter surprise, they found that C3H2O is different. C3H2O is different. I have highlighted, it's different from its parent molecule C3. All these years, scientists never carried out spectrophotometry. No idea did not strike them. Once they did, they found that the spectrum of C3H2O was different from C3. And then once the difference was sorted out, then they carried out a quantification. Again in quantification they found C3H2O was just 0.5% of the C3. 0.5% was C3H2O, remaining 99.5% was C3. So, C3 was the majority, C3H2O was minority, just 0.5%. Nevertheless, this 0.5% was very different from the parent molecule C3. So, new wonders of science was expressed. C3H2O in the presence of magnesium ions Mg2 plus, they would bind to the microbial surface via thioester linkages. This was elucidated. So, we want C3H2O to bind to the microbial surface. Once it binds to the microbial surface, the alternative Tickover pathway starts. Alternative Tickover pathway, in very simple terms, you try to understand that alternative Tickover pathway uses 1 plus 1 equal to 2 C3, I am abbreviating it as CONV, that is 2 C3 convertases, and those two C3 convertases, which I will be explaining in a classical Dr. Raktim Mukherjee style of lecture, is one is fluid phase C3 convertase and another is membrane bound C3 convertase. Okay. I will just use my eraser, erase everything, because I will come back to this slide after I explain all this in my own style. My own style first let me explain, then I will come to textbook style. So, I go to the next slide. Okay, everything is animated, so it will be very easy for you to understand. Full attention. Okay, stay focused. This can be a possible 2-mark question in examination if asked. Formation of fluid phase C3 convertase. But I am 100% sure people may not go into this much depth in other colleges or even in your department. But we should all study, because we are science students, we cannot leave anything. So, how fluid phase C3 convertase is formed, that is our objective. So, let us form fluid phase C3 convertase. The first player is C3, as the name suggests, C3 convertase. So, the first player has to be C3. So, you have C3. I request students, please stop me, it is a live online lecture. Wherever you do not understand, please stop me. So, fluid phase C3 convertase, it starts with C3. Remember students, we talked about Tic Tac, right? Not TikTok, which is a Chinese app that is banned. Tic Tac chocolates are available in D-Mart. You just open the lid, one chocolate you take in your mouth, again open the lid, and every time you open the lid, a specific sound comes, the Tic Tac. So, this also C3 undergoes spontaneous hydrolysis, continuously it undergoes hydrolysis. Tic Tac, it keeps on undergoing hydrolysis. So, C3, once it undergoes spontaneous hydrolysis, it yields something which you saw with spectrophotometer in the earlier slide, C3H2O. I hope first step is clear. C3 undergoes spontaneous hydrolysis in fluid phase within water or aqueous phase to give C3H2O. This is C3H2O, which is very different from the parent molecule C3. Now, what happens? You have serum magnesium, magnesium ion present in the serum. This serum magnesium will closely link factor B along with C3H2O. Now, what happens? There comes the next molecule factor D. This factor D would break factor B into two parts. It would break factor B into two parts. The first part being capital B small a, red color I have highlighted is, it is a reddish pink I have highlighted, it is a smaller molecule. Being smaller molecule, it would diffuse away, whereas capital B small b is a larger molecule that is going to remain. So, what happens to this smaller molecule? What happens to this larger molecule? Let us go ahead. Capital B small a being the smaller molecule, it diffuses away. So, with PowerPoint I have just diffused away, it goes away, whereas capital B small b remains, and this capital B small b that remains will form along with C3H2O, no, because this was already there, it binds with C3H2O to form fluid phase C3 convertase. Someone asks you in examination how fluid phase C3 convertase is formed. These are your steps. You have C3 undergoing spontaneous hydrolysis. Spontaneous hydrolysis of C3 gives rise to C3H2O. In the presence of magnesium ion, C3H2O docks with factor B. Factor B is acted upon by factor D. This factor D breaks factor B into two unequal fragments. One fragment is factor capital B small a, the smaller fragment which diffuses away, and the larger fragment remains. This larger fragment joins with C3H2O to form fluid phase C3 convertase. I hope. Now, our focus shall be on membrane bound C3 convertase. One of the highlights we said of alternate pathway, alternative pathway has two C3 convertase. One was fluid phase C3 convertase, just now we discussed. The next one is membrane bound C3 convertase. How membrane bound C3 convertase is formed, that shall be our second part of discussion. So, again, as the name suggests, C3 convertase, obviously whenever we talk about C3 convertase, things should begin with C3. So, we have C3, as the name suggests, C3 convertase. So, C3 is acted upon by fluid phase C3 convertase. What is fluid phase C3 convertase? Just now we saw C3H2O capital B small b is the fluid phase C3 convertase. So, can any of my students say what C3H2O capital B small b is going to do to C3? What would they do?
It will break into two parts, unequal parts.
Which two unequal parts? Which is smaller, which is larger?
C3A is the
C3B is larger.
Okay, fair enough. Let's see. So, it breaks C3 into two unequal fragments, C3A the smaller fragment and C3B the larger fragment. To summarize, C3 under the influence of fluid phase C3 convertase gets broken down into a smaller C3A fragment and a larger C3B fragment. We repeat the steps once again with a new perspective. Now, C3B being the smaller, A being the smaller fragment diffuses away, and then factor B gets a chance to bind with C3B. Once factor B binds to C3B, factor D will get a chance. So, factor B will bind to C3B. Together, factor B and C3B together, they forms a complex. That complex is called as C3B factor B complex. Now, what happens? Like the previous step, that is C3 convertase fluid phase, this factor B will be acted upon by factor D, and this factor D will break factor B into So, factor D breaks factor B into two unequal fragments, and those unequal fragments are capital B small a, the smaller fragment, and capital B small b, the larger fragment. As the name suggests, capital B small a, the smaller fragment diffuses away. What is left with us is capital B small b, and this leftover fragment would combine with the previous steps C3 to form C3b capital B small b, and this is student called as membrane bound C3 convertase. Membrane bound C3 convertase. I hope for the entire class two set of instructions are complete. First, how fluid phase C3 convertase is formed, and next how membrane bound C3 convertase is formed. I hope you have understood both concepts, everyone.
Yes, sir. Yes, sir.
I am deliberately going very slow so that it can hit your brain and you can understand. Next, if you have understood this much, rest of the things are very easy in fact. Then we come to the next part, C3b acted upon by factor B, factor D breaks it, some repetition, factor BAB and BB diffuses away. C3 acted upon by C3b capital B small b, this is nothing but the C3 convertase. There are two C3 convertase, one is fluid phase C3 convertase, and this C3b capital B small b is membrane bound C3 convertase. Membrane bound C3 convertase, as the name suggests, C3 convertase, membrane bound, it will act on C3, and this C3 will be broken down into the C3a and C3b. C3a being the smaller fragment diffuses away and C3b remains. This C3b that remains will not act till it is stabilized by properdin. Very important MCQ question, which molecule stabilizes C3b? C3b formation, student, is an amplification step. So many molecules of C3b is formed, but if there is no pathogen, this C3b will be degraded, but if C3b deposits on the surface of pathogen, the properdin will stabilize C3b so that it can go for long action. That is, C3b will be pulled by, or it will be concentrated by properdin. Properdin stabilizes C3b. So, which molecule stabilizes C3b? Properdin stabilizes C3b. So, now next step, C3 is acted upon by C3b capital B small b. This is nothing but C3 convertase of alternative pathway. Students, please note, the convertase of classical pathway is very different from that of alternate pathway. Can any of So, now comes the next part. So, this C3b and C3b of the properdin pathway, they together will together form C3b capital B small b and yet another C3b, this one, yet another C3b. So, together they form C3b capital B small b and C3b. This is nothing but the C5 convertase of alternative pathway. Remember students, even the C5 convertase of alternate pathway is different. What was the C5 convertase of classical pathway? Anyone?
C4B, 2A, 3B.
C4B, 2A, 3B. That's fantastic. And here in alternative pathway, the it is C3B, capital B, small b, and C3B. So that forms C5 convertase. As the name suggests, C5 convertase, it will convert C5 into two fragments, C5A and C5B. It will form two fragments, C5A and C5B. Remember students, C5A is a potent anaphylatoxins, very important role in generating inflammatory response. So C5 under the influence of C3 convertase will break down into C5A and C5B. A being the smaller fragment diffuses away, being the larger fragment remain. Then there is assembly of C6, C7, C8, C9 complex. This complex has unstable membrane binding site. So they further combine with C6 and C7 together with C8 and C9 to form the membrane attack complex. So this membrane attack complex, also called as the MAC, the membrane attack complex. This membrane attack complex is the one like a drilling machine. It is going to drill a hole on the pathogen cell, and through that hole it will undergo lysis or breakdown. So then came a fantastic research paper. I just said that when I was teaching my first batch of students in 2008, this research paper had come. Properdin binds to late apoptotic and necrotic cells independently is very important. Of C3B, C3B just now we saw, and regulates alternative complement component activation. So students, now according to research, three pathways are there. One just now we completed, the lengthiest one, alternative Tickover pathway. Now we will discuss properdin pathway. The problem with properdin pathway is, under in vivo condition, properdin pathway is not known to act, but under in vitro condition, it is very prominent. Independently, it can activate the complement pathway, that is also called as properdin-activated complement pathway. So alternative properdin-activated pathway, this is the fundamental principle. Properdin, it will stabilize, and then you have C3B and factor B joining together to form properdin C3B capital B's complex, and then factor D comes, it will break factor B C3B capital P is properdin capital B, and then like the previous pathway, factor D will then break capital B into BA and BB. But what interesting I am trying to convey student is that under in vitro condition, even properdin alone can initiate or start or activate alternative pathway. So this is the latest research. Otherwise we already knew, even in 2008 when I taught, that properdin, what properdin does, properdin stabilizes the properdin it stabilizes the C3B. How it stabilizes? How it stabilizes, let's see. Properdin can directly bind to the surface and can recruit C3B and factor B. factor D is recruited and cleaves factor B into two parts, capital B, small b, and the resultant C3b, capital B, small b is an active C3 convertase. What is the biochemical nature of C3b, capital B, small b? This is the active biochemical convertase. It is an active C3 biochemical C3 convertase. The next part of the story is alternate protease-activated pathway. Protease-activated pathway. The alternative protease-activated pathway also is not. It's a very recent topic that has come. Initiation of clotting cascades have been shown to stimulate cleavage of complement proteins. It will initiate a clotting cascade, and that has been shown to stimulate the cleavage of complement proteins. Students, this protease activated pathway you might have heard in terms of blood clotting during clotting factors, the same protease activated pathway. This protease, especially thrombin, can activate the alternative pathway. So, thrombin cleaves C3 and C5 in vitro condition. Platelet activation releases ATP, calcium ions and serine threonine kinase, and all this could stabilize C3 in the fluid phase. And this indicates, since C3 and C5 are cleaved, what is the product of C3? C3a and C3b. What is the product of C5? C5a and C5b. So, this C5a and C3a, they are potent anaphylotoxins. They indicate a strong inflammatory reaction that could be potentially activate the complement system. So, therefore, the strong inflammatory reactions could be potentially activate the complement system. Now, sometimes even under clinical setup, just now D-dimer internal clots in COVID. So, whenever the complement pathway gets activated to protease pathway, or whenever there is a blood clot which activates the complement pathway, it can be catastrophic. But from academic point of view, it is important for you to understand, students, that alternative pathway can be activated by three means: continuous Tickover hydrolysis to form C3H2O from the protease activated pathway, as well as from the properdin activated pathway. So, three ways of alternative pathway getting activated. Now, some of the questions to end this segment. Just right answer. Next question. Addition of which of the following components to any C3 convertase leads to the formation of C5 convertase? C3b, C2a, C4b, C5b. Students, all these are entrance examination questions. This time I am really impressed with your class. Everyone has gone with option A, and yes, option A is the right answer. C3b, right, they form C3 convertase. So, option A is the right answer. Thank you, students, for your kind attention. So, that concludes the first 40-minute session on alternative pathway. APMS campus offers 60 plus courses across 22 institutes, right from mathematics, physics, chemistry, biological sciences, medicine, that is occupational therapy and medical lab technology, you please visit our website for further details of the courses. As far as biological sciences is concerned, Shri P.M. Patel Institute offers master's program in microbiology, biotechnology and biochemistry. Join APMS campus and add wings to your academic thoughts.